event. It's a real pleasure to be with you and an honor for both me and for Pfizer to be invited to speak. It's interesting that an important day for year for when we think back, it was 60 years ago this year that Pfizer Taiwan was established. Over those 60 years, we've worked closely with the Taiwanese government and Board of Health, with opinion leaders, experts like yourselves, with physicians that are doing to the people of Taiwan. I think that's something that we all take as a very important responsibility. And we look forward not just to another 60 years, but another 600 years to continue that process of making the lives better of people in Taiwan and around the world with life-saving drugs. So it's a pleasure to be with you. So I want to start with my disclosures. I'm a full-time employee of Pfizer, as you know, but this presentation will be non-promotional. I'm going to try to avoid even speaking to specific molecules because I don't want this to be a promotional piece. We're talking about at a higher level. In terms of abbreviations, we often use AMR when we're speaking about antimicrobial resistance and AMS to refer to antimicrobial stewardships. Those may come up as we're speaking. The agenda today is to start with an AMR perspective. This is important to understand what the environment was like when SARS-CoV-2 decided to infect humans. And then there's three questions I've been asked to discuss: how COVID-19 impacted the pharmaceutical industry and multi-drug resistance. What are the stories behind the difficult decisions that were made, the breakthrough that Pfizer has achieved in recent drug development? We call it our Lightspeed program. I grew up, I hate to admit this, on watching Star Trek as a child. And so Lightspeed has a very special meaning to me. And it has a very special meaning for Pfizer. It's doing things faster than we ever thought possible so that patients can benefit from life-saving therapies more quickly. And then lastly, how will Lightspeed practice potentially affect the industry and clinical practice in the future? So let's start at the beginning. We all know about Sir Alexander Fleming in 1928, who discovered penicillium, producing a substance that at the time was, we were uncertain what it was. We knew that this large colony of penicillium was secreting something into the agar that prevented the staph colonies from growing as large if they're close to the contaminating mold. As we all know, that turned out to be penicillin, which was described as a wonder drug from mold. You know, in this day and age, it's hard to remember the impact that penicillin had on patients. This is a graph published the year I was born that showed the patient survival with pneumococcal bacteremia. In nearly all cases, I would have expected these patients started with a pneumococcal pneumonia that spread to the bloodstream. And we know that without therapy, 80% or so would die from that infection within 16 or 17 days. It was found that if we transferred serum from a patient that survived to a patient that was currently infected, we could improve survival markedly. We found that survival was slightly less than 60%. If slightly less than 60%, if we transferred the immune factors that were in the serum. But penicillin was revolutionary, and you see this at the top. Nearly 90% of patients would survive this devastating, life-threatening infection with an antibiotic, with penicillin. So let's think more broadly now with our more current antibiotics. This is a table that was developed by David Livermore back in 2019, and it remains fairly accurate. It looks at the likelihood of death in the pre-antibiotic era for a number of diseases and the likelihood of death in the post-antibiotic era with the same diseases. And what we found is a marked impact due to antibiotics. Skin infections. It's hard to think of a time when 10%, 11% of patients with skin infections would die from that infection. Even those that survived, a very high percentage would lose feet and hands, limbs, arms to try to prevent that infection from spreading. So even if they survived, they often had very devastating sequelae. Nowadays, in the era of antibiotics, less than 1% will die from all type skin infections. Endocarditis and brain infections used to be almost uniformly fatal. Today, roughly 20% of individuals will die if they're infected, if their heart's infected, and less than 20% with brain infections. That's still too high, but it's a whole lot better than how it used to be. So there's still work to be done, but antibiotics have made a huge impact, especially when we compare those changes, those life-saving percentages, to something that we also think is important, common. Someone who has a myocardial infarction is always treated with aspirin or streptokinase, a clot buster, because we know that improves life expectancy. But when you compare that to antibiotics, it saves about 3% of patients that otherwise would have died. Now, if I have the if I'm having the heart attack, take the 3%. I like 3% as opposed to 0. But still, I think this emphasizes the revolutionary change that antibiotics have had on medicine. And that's allowed the development of modern medicine. Because we have effective therapies, we can treat patients for cancer, suppress their immune systems for the time. We can do surgeries that we could never do before because now we have antibiotics and can keep those patients alive. Chronic conditions such as diabetes, the renal failure that requires hemodialysis, or hardware replacement, hip replacement, knee replacements, AV fistulas, all feasible now only because of effective antibiotics. If we did not have effective antibiotics, the risk would be simply too high to do many of these things because an infection would certainly be lethal without effective antibiotics. So we often think of antibiotics as the foundation of modern medicine. We don't think about that perhaps enough, but without effective antibiotics, much of modern medicine could not be performed. This even came to the point where in 1967, William H. Stewart, then Surgeon General of the United States, said, and there's some controversy about really whether he said these exact words or not, but there's no question that the thought was prevalent in American society, is that it's time to close the book on infectious diseases and declare the war against pestilence won. The picture of the polio ward is to remind us it wasn't just antibiotics he was speaking to, it was also vaccines, and the polio vaccine had really removed the threat of the sequelae of polio from our society. It's interesting to look back and think how wrong they were, and I think everyone in the room would kind of chuckle at that statement. But it did cause some changes to occur. We saw, and this is from the FDA Food and Drug Administration, the National Research Council. We saw this innovation gap. People stopped doing research on antibiotics. Similarly, a discovery void was noted, where again, just the research stopped being done because it wasn't felt that it was needful. Even now that we know it is needful, that there is this need, there's still barriers to antibiotic research. And this slide's a good one from BCG analysis, which shows that the difference between antibiotics and other therapeutics. So if we think about a new diabetic medication or high blood pressure medication, there's lots of people with both of those illnesses. And so if you had a new drug that was better, superior, worked better, that innovation would drive sales. You would have a lot of sales because all of those people would want to get on the best drug. And also it would allow for a reasonable price, leading to a predictable and a high revenue, so that companies would be interested in doing that research. Antibiotics are different, and I think we recognize that. A very innovative antibiotic that addresses really new, interesting multi-drug resistant organisms, the use is limited only to those patients that need the drug, and rightly so. I agree that that's what we should be doing, but that creates a very small pool of patients that are going to get that drug, at least initially. We also have this perception in societies around the world that antibiotics should be cheap, that they're just a drug that we don't charge much for. That combination leads to unpredictable and low revenue. And let me show you a little bit more how that works out. This is a table created by BCG Analysis and Evaluate Pharma. And what it looked at is the cost of developing drugs in different classes. The red box shows how much it costs roughly to develop that drug. So if you look at an oncology drug, approximately $450 million to identify the drug and develop it. Remembering that a lot of those drugs will get partway through development and will fail. So on average, overall, it takes about $450 million. But above the line in the green color shows the amount of revenue that companies expect to gain from that drug if it is successful. So if you look again at oncology, it may cost $450 million to bring to market, but the revenue on that is expected to be $8.2 billion. The return on investment is reasonable. You can look at the other areas, oncology, respiratory disease, dermatology, musculoskeletal, but look at antibiotics. It costs about $500 million to develop an antibiotic. But in terms of the net present value, that money is not returned to the company at the end. This is important and noted by the lay press. This is Bloomberg Business Week that talks about antibiotics are not profitable enough for big pharma to make more. And so many pharma companies left the field. This is just a list that I've created personally of a number of companies. The medicine company abandoned their antibiotic R&D effort after getting approval for Vabomere. Then they sold that asset to Melinta. Novartis and Sanofi and Novartis abandoned their antibiotic research and development efforts, divested their clinical and preclinical assets. Allergan is one that's kind of is sad that they were holders of North American rights for ceftaroline, dalbavancin, ceftazidime, avibactam, some of our best antibiotics, but announced that they would divest that antibiotic, all of their antibiotic assets. Melinta, remember, that purchased Vabomere from the medicine company, was the largest pure play antibiotic company in the world. They had delafloxacin, meropenem, vabobactam, oritavancin, and IV minocycline. They abandoned all of their R&D effort before filing for Chapter 11. And you probably looked through the rest of this list already. The point is, companies were exiting the field of antibiotic research. This is from the Pew Trust Research Center, where they noted that between 1990 and 2016, the number of large big pharma companies active in antibiotic R&D fell drastically, from 18 to only 6 in 2016. It's probably, and it would be, that number would be lower now. I'm proud to work for a company that remains in the field of antibiotic research. It's an important area. We recognize that, and Pfizer is committed to continuing. So that's the environment. So along comes SARS-CoV-2 with that understanding. Fewer big pharma companies invested, most companies feeling like it's not a money-making area and they don't want to invest money into it. And then in December of 2019, we had those first cluster in December, end of the year, that first cluster of patients in China that had SARS-CoV-2. And we know what happened after that. It's a difficult time. It would have been better had SARS-CoV-2 came earlier when more companies were involved in research, no question. But it wasn't as simple as that, was it? So we know that all of us have been infected with coronaviruses in the past. They tend to be mild. They move through schools, through daycare, through families, generally causing a cold-like syndrome. And all of us, in all likelihood, have been infected by at least one of the coronaviruses when we were children or young adults, or even sometimes as an adult. And then we saw SARS-CoV-1. That was the first time that we really recognized that coronaviruses could cause a significant infection, not just a cold, but something far more serious. And you know that between 2003 and 2004, SARS-CoV-1 infected approximately 8,000 people from 29 countries, resulting in at least 774 deaths. Now, Pfizer had in the past, had a viral program looking at some of the common viral illnesses and developing molecules that could address those. Included in that list was coronavirus. There was not an optimized product, an optimized molecule from that research effort, but nevertheless, we had molecules that we had identified as being active against coronavirus. So when SARS-CoV-1 hit, we pulled those molecules out of the freezer and looked at asked the question, were they active against SARS-CoV-1? But remember, that infection burned out very quickly. It ceased to cause a global problem, so Pfizer put that research on hold. Then we saw MERS-CoV, right, which caused Middle East Respiratory Syndrome, MERS, between 2012 and 2015. And again, in that case, about a little less than 2,600 patients were confirmed, with slightly less than 900 confirmed deaths. But again, that hit and disappeared. And so not a lot was done. Then came SARS-CoV-2. And remember, first reported in December 30th of 2019. Pfizer started their phase 1 study on the prioritized, the best molecule that appeared to be active against SARS-CoV-2 in March of 2021, which is really pretty remarkable considering that the first reported case of SARS-CoV-2 was identified at the very end of December in 2019. Very, very rapid. The phase 2, phase 3 study started in July of 2021, and then emergency use authorization occurred December 21st, which was only 2 years, almost perfectly to the day, 2 years after the first case was identified. Now, Pfizer played a big part in how fast that occurred, but it wasn't just Pfizer. It was patients that were willing to enter those studies very, very quickly because of the death, because of the fear. But nevertheless, anytime a patient enters a study, they're doing a service to their fellow person, to their family members, to the future society, and to their community. And so they were a big contributor. Also, governments would review the data very, very quickly and move forward quickly because they saw the need, just like Pfizer did. So I want to talk a little bit about some more detail in that area. So if you think about it, right at first when SARS-CoV-2 struck, when we started to see cases, it was unclear if it was going to burn out like SARS-CoV-1 and MERS or whether it was here to stay. And it really wasn't until somewhat later that we started seeing significant number of cases that told us that yes, indeed, SARS-CoV-2 was going to be a problem. So this is, and for folks that aren't aware of how drugs are developed, this gives you a basic idea of how it is developed and the basic timelines. The basic research is identifying the molecules that are active against that pathogen. Then it moves to discovery research, often animal research where we determine if the drug is toxic to mammalian hosts and also whether it's effective in models of the disease in animal models before we move to humans. It also is the time that you optimize your drug. You may have 5 or 6 drug candidates that could potentially address the pathogen, but you have to pick one to move forward. And so this is a time where you look at your animals and determine which drug is not going to be toxic, which drug is going to have the best efficacy to pick the molecule to move forward. And that can take between 3 and 6 years. Once the molecule has been chosen, then we move to human work. And the phase 1 study, we also call it first in man, would be single dose studies to look at toxicity, and then multiple ascending dose studies in healthy adults. Phase 2, we go to volunteers that have the illness, and we look to see how the drug works, how the patient responds with active infection. By the end of phase 2, we like to have what we call proof of concept, evidence that the drug really is effective and really having an impact. The reason for that is phase 3 studies tend to be much larger, much more expensive, and take much more time. So we like to have a good idea that they're going to be, that there's a good chance that they're going to be useful and that the drug's going to be useful before we invest the time and the money in doing further research. Again, that's in sick patients, volunteers, and you look at efficacy and then show efficacy. And then after you finish your phase 3 study, that's when you get your market authorization. But it can still be, half a year to even 2 years before you ramp up drug supply and that you're able to get it to patients and you actually start getting market access. So that's the way it's normally done. And I want to compare that, or use as a comparison, my academic career. So like most folks in the US, I did 4 years of an undergraduate degree before I entered a program. Mine was a dual MD PhD program, 3 years in the MD, 5 years in my PhD, and then I did a year of postdoc, and then 3 years in an internal medicine residency, and then a fellowship in infectious disease. Looking back, it's hard to believe that's a total of 20 years of my life was spent between starting undergrad and finishing my infectious disease fellowship. But what if I could have done that different? What if I could have done something like this? So started my MD during my undergraduate years, to cutoff years, to do my postdoc during my PhD year, doing my internal residency perhaps at the end of my PhD year, and overlapping my residency and my fellowship. Potentially could cut down the time it took me to get from one end to the other quite significantly. And I know we all kind of chuckle at that. That can't be done. It has to be done sequentially. But think about it if you could. What if you could do that, but they told you you need to pay for this next step before you know if you can use the next step? So before you know that you've graduated from your undergraduate time, that you have to pay for your MD. Most of us would probably pause and give that a little bit of thought. So let's translate that now into drug development, especially now that we've talked about drugs are development. And remember again, phase 1, phase 2, and phase 3, and then a gap between phase 3 generally before you have enough drug to release it to the market. All right. So here's how it's typically done. The preclinical studies that would include animal tox in 2 species and genotox, reproductive tox, there's more studies in there, but those are some examples, would be completed. We would know that the drug is safe, at least based on our animal work, before we start the phase 1 studies, the first-in-man studies, before we set those up and progress. That's how we normally do it because that minimizes the risk. You don't spend the money until you know that you're going to, in all likelihood, be able to use that money, that it will have a good impact. And what we did for both our small molecule and for our vaccines was to change that to some degree. We started site selection and inspection and training in the phase 1 studies while these animal studies were still ongoing, while the preclinical studies were ongoing. Now, invested that money. Now, you don't start giving the drug to patients until you have the preclinical data done, completed, evaluated, and you know that it's safe. But there's so much of that preliminary work that could be done at risk, recognizing that if some point in this preclinical work, if we had found that the drug was toxic to an animal, that program would be canceled. And all of the money spent on the site selection, inspection, setting up the study would be a loss to the company. So there's risks to that, but the advantage is you're able to compress those timelines and move through more quickly. Let's give another example. I think this is also a good example, and one that it's hard to convey how amazing I found this. This is not what my area of work, but it's amazing when I've learned of it. So remember, you start drug synthesis early. You have to have enough drug for your animal studies and for your phase 1 studies, your phase 2 studies. During this time, phase 1, phase 2, you're wanting to be sure that your drug synthesis pathways are the pathways that you're likely to use for your big clinical study, your phase 3, and then subsequently when you start to market your drug. So there's many, many steps here. There's many refinements, with the idea being that drug synthesis increases very slowly. You need more drug at each step, but it's a slow increase. And then once you get to, my little box is in the, there you go. Once you get to approval, then generally companies will really ramp up production. And that's why you often have a 6-month to even up to a year, year and a half, 2 years gap between approval and real market entry, because that's the time that companies use to ramp up drug production. But what if you did this instead, which is exactly what Pfizer did? You ramp up that drug synthesis much more quickly so that the supplies are much more robust through the process. Again, there's risk to that. If the phase 3 study was a failure, all this additional work on drug synthesis would have been lost. It would have been wasted drug, wasted effort, wasted money. But the advantage to that, as you see here, and this is all in that Pfizer announced publicly, the first 180,000 doses of our small molecule to treat SARS-CoV-2 was shipped actually in 2021. Remember, it was only approved late in 2021. So 180,000 doses were already waiting, already packaged correctly with the right labels, and were shipped out within the last few days of the year. As of the end of May 2022, Pfizer had shipped more than 12 million dose treatment courses of our small molecule to nearly 40 countries around the world. That's remarkable. 12 million doses within 4 months of approval. I can't think of another time when that occurred. Maybe somebody could give me an example, but I certainly can't think of one, which really, again, demonstrates that at risk, the work that was done to try to ramp up drug synthesis. And remember, it's not just the factories where the drug's being created. It's not just where it's packaged, but it's the molecule, the chemicals that go into the synthesis. Most of those are in limited supplies around the world. Oftentimes they're being used for other things, other molecule creations, other drug creation. And so it's difficult to get access to those drug supplies quickly and easily. And again, Pfizer went to great efforts to try to secure those supplies early because we knew if the drug worked, patients were going to need it right away. We can't wait 6 months, a year, 2 years to get a life-saving drug to the patient. That rate of synthesis is increasing dramatically through 2022, and there's different targets for how many doses, actually courses of the drug that will be shipped by the end of the year. But it could approach 100 million courses of drug, which is remarkable. And again, makes me very proud to work for Pfizer. So those were a couple areas where we were able to at risk invest and speed this process. But the other question I was asked was, what about the impact of antimicrobial resistance? We know that COVID is accompanied at times with co-infections of bacteria and fungi. Oftentimes when a patient's very ill, it's difficult to know what's actually causing the respiratory failure. Is it SARS-CoV-2 or is it a bacteria? And so we know that antibiotic use did increase. This is a headline from the Washington Post: pandemic fuels surge in superbug infections and deaths, CDC says. There's a whole report, and this is the reference at the bottom, that the CDC has put out of the significant rise in antimicrobial resistance. They attribute that increased use because of sicker patients, antibiotic overuse, staff and equipment shortages. In many cases, patients are cohorted much more closely than they otherwise would. The need for isolation is much more significant with SARS-CoV-2. But nevertheless, we've seen this impact. So SARS-CoV-2, we think about the importance of SARS-CoV-2, and we should, but SARS-CoV-2 is also changing the landscape. And if you think back to the first part of the talk, that's a concern. More companies need to be involved in doing the work. Because if we allow the foundation of antibiotics to fail, if we stop having effective antibiotics that work, we see a collapse of modern medicine. Suddenly it's difficult to immunosuppress patients with an anti-cancer drug if we know that the risk of succumbing to an infectious disease is just too high. Surgeries become much more risky, chronic conditions more difficult to treat, and hardware replacement becomes much more risky. And also, we have to remember we're not out of the woods. This just came out 3 days ago now in The Guardian, but just reminding us that new variants, subvariants of COVID will be coming. COVID is an RNA virus. RNA viruses are good at mutating, so that's not unexpected, but nevertheless, there's more to do for all of us. And as I said at the beginning, it's not just Pfizer company that does it. Governments, boards of health, patients, physicians, we have to work together to solve the problems. Otherwise, we have a possible future that was described by David Cameron in 2014, that if we fail to act, we are looking at an almost unthinkable scenario where antibiotics no longer work and we're cast back into the dark ages of medicine. And that's my last slide. Again, it was a real honor to speak to you all. I wish I could have been there in person. And it looks like I am right on time. So I turn the time back to the chair. Thank you.